And good afternoon, and welcome to Jason's thesis defense. And before Jason starts his defense, as his thesis advisor, I would like to say a few words about him. And first, a brief introduction about Jason. And Jason received his bachelor's degree in statistics from Fudan University, which is a top university in China. And in 2013, he joined us and became a SCM student in our department. And 2 years later, in 2015, as one of our top SCM students in his class, he was admitted to our PhD program. And not long after he joined us, many of us realized that he is really a special student. And as his advisor, I have truly enjoyed working with him for the past 7 years. And Jason has a long list of accomplishments as a student. And as you can see from his CV, as a master's student, he has won our Kocha-Lakota Award, which recognizes his outstanding performance on the SCM comprehensive exam. And when he was a master's student, he already has a first author paper accepted. And as a PhD student, he has won our departmental Junkali Award for his outstanding achievement on the school-wide examination paper. And he has also won our Margaret Merrill Award, which recognizes his outstanding research as a doctoral student. And he has led a team to participate in the ENCODE Dream Transcription Factor Binding Sites Prediction Challenge, which is an international competition, and won the 4th place out of the 50 international teams. And he has also won 2016 Distinguished Student Paper Award by the American Statistical Association section on statistics in genomics and genetics. And shortly after he started his PhD study, he had started work on an exciting new topic that is single-cell genomics. And this is an emerging new field. And just within 3 years, he has already made important contributions on developing statistical and computational methods for analyzing single-cell genomic data. And his T-scan method was one of the best performing algorithms for single-cell pseudotime analysis. And his SCRAD and SCAGE software for analyzing single-cell atype-seq data are among the first of such tools. And he also played crucial roles in many of our collaborative projects. For example, in one of our collaboration with Dr. Kelly Smith, who's in the committee here, and also Dr. Drew Pardo, Jason is a key person for analyzing their coupled single-cell RNA-seq and single-cell T-cell receptor sequencing data. And Jason has been highly productive. He has published 17 papers so far, and this includes 6 first author papers. And he has also developed many software tools, including some of the very popular tools in single-cell genomics, such as T-scan. And this year, Jason was on faculty job market, and he got offers from both Emory University and Duke University, and was also considered by the Fred Hutchinson Cancer Research Institute, Cancer Research Center. And eventually, he decided to join Duke as a tenure-track assistant professor in biostatistics and bioinformatics, and also as a member of their interdisciplinary quantitative immunology program. And one special trait of Jason, which I think all of my collaborators will agree, is his unbelievable efficiency. And if you give him a task and ask him, will you be able to get the results to us in 10 days? His reply is always, no problem. I can get the results tomorrow. That's Jason. And I know I definitely will miss him after he leaves my group. And but I'm happy that he will become an independent investigator. And anyway, Jason, it was a truly wonderful experience working with you. And I'm sure you will continue to do great things at Duke. And assume you, of course, that's by assuming that you will pass today's defense. And I look forward to celebrating your many accomplishments in the future. And without further ado, let me turn it over to Jason. Jason, it's yours.
Okay, so can you see my screen and hear my voice?
Yes, I can see your screen.
Okay, okay, thank you. So thanks, Hongkai, very much for the wonderful introduction. Let me see. Okay, so it's a great honor for me to present some of my research work during my PhD studies today. And today I'm going to introduce some statistical methods we have developed for decoding gene regulation in single cells. So single cell sequencing is a transformative technology that measures the genomic information in individual cell. It was named as a Method of the Year by Nature Methods in 2013 and 2019. Single cell sequencing technology has been evolving rapidly. The number of cells in a study increases from less than 100 cells a decade ago to almost 1 million cells in recent years. Why people are so interested in single cell sequencing? This is because single cell sequencing measures the genomic information for each individual cell, which often coming from a heterogeneous cell population such as cancer. It enables the identification of rare cell population as well as a comparison between cell subtypes that usually cannot be done by traditional bulk sequencing that only measures the average genomic information for the whole cell population. Single cell sequencing has been widely applied in biomedical research, such as the study of immune cells in tumor microenvironment, the developmental lineage of a whole animal, the spatial transcriptomics of mouse brain, and identifying new cell types in mouse kidney. These are just few examples, and there are many more. Single cell sequencing also made it possible for the first time to create comprehensive reference maps of all human cells, which is a mission of a large ongoing international collaboration project called Human Cell Atlas. Its goal is to sequence at least 10 billion cells, covering all organs, systems, and tissues in human. Once completed, it will serve as a basis for understanding human health and treating diseases. We are also conducting many research projects related to single-cell sequencing, such as identifying a transcriptional network in T-cell exhaustion process, inferring the tumor origins for glioblastoma patients, finding the molecular signatures in a neoadjuvant immunotherapy for non-small cell lung cancer patients, and studying the changes of regulatory activities in pancreatic metastatic cells during drug treatment. The central theme of all these research projects is a study of gene expression and how gene expression is regulated. Gene expression is an essential process where DNA is made into functional gene products of RNA and proteins. And this process is controlled by a wide range of sophisticated mechanisms, which ensures the cell has the right amount of gene products. Why are we interested in gene expression? This is because in our human body, although almost all cells have the same DNA, their functions can be very different. This is largely due to the fact that different sets of genes are being expressed and different gene products are being made to support the different functions of the cells. Genes expressed at wrong time, wrong place, or wrong amount may lead to serious consequences such as developmental disorder or cancer. Thus, understanding when, where, and how much a gene is expressed, as well as how is gene expression regulated, is essential for understanding the mechanism of these complex diseases, as well as providing guidelines for future clinical treatment. In today's talk, I'm going to tell you two stories as how we combine new statistical methods with data from different single-cell sequencing platforms to help us better understand these two questions. In the first part of my talk, I'm going to introduce single-cell transcriptome analysis, where we study the expression of each gene in each cell. There are many issues in this field, and today I'm only going to introduce one specific issue, that is how to use the cells from a sample to infer its underlying biological process. To address this issue, we developed a new method called T-scan that performs a pseudo-time analysis with cluster-based minimum spanning tree. As a motivating example, I'm going to show you a single-cell RNA-seq dataset from Human Cell Atlas. This dataset contains gene expression profiles for more than 20,000 genes and 200,000 bone marrow cells. Single-cell RNA-seq data is highly sparse and noisy, and it takes a lot of time and effort to adequately pre-process the data. We have also done much work comparing and benchmarking different pre-processing methods. For example, we have just published a preprint on bioRxiv comparing the performance of different single-cell RNA-seq imputation methods. Since the pre-processing is not the focus of my today's talk, I'm going to skip this part, but you are welcome to discuss with me after the talk. After pre-processing the data, we can map the cells on a low-dimensional map. Here, each dot is a cell which is essentially a high-dimensional gene expression vector with the expression values of more than 20,000 genes. We can see that there's a pattern of 3 branches originating from the center of the plot. And you may wonder what this pattern is. After computationally identifying cell types that are marked by different colors on the plot, it turns out that this pattern actually agrees with the process of hematopoietic stem cells, or HSCs, differentiating into myeloid, erythroid, or lymphoid, which are major blood cell types. This is a process people already know, so it suggests that the single-cell RNA-seq data contains the information about the underlying biological process. However, in many situations, people don't know about the underlying biological process, and the goal of single-cell sequencing is to infer this unknown process. So it raises the question as whether we can use the observed single-cell sequencing data to infer the unknown biological process. That's the issue pseudo-time analysis tries to address. A pioneering work done by lead author Trapnell and his colleagues in 2014 is called Monocle. Here shows the same low-dimensional representation of the cells, and the Euclidean distance between 2 cells on this low-dimensional representation actually corresponds to the similarity between 2 cells. If the 2 cells have a smaller Euclidean distance, then they are more similar to each other. Monocle uses a minimum spanning tree to connect each individual cell. A minimum spanning tree is an undirected and acyclic graph that connects all cells and has a minimum total edge weight. Here, edge weight is defined as the Euclidean distance between 2 cells on the low-dimensional map. After that, Monocle tries to find the longest path through the minimum spanning tree, which is marked as a red line here, and project each individual cell onto the red line to form a trajectory. And the position of the cell on the trajectory is called pseudotime, which reflects the state of the cell transitioning from one cell type to the other. It is called pseudotime because it is not the real biological time, but rather an estimate of it. Once we have the pseudotime, we can study the expression of each gene along pseudotime. For example, we can study the expression of a gene called HBA2, which is a non-marker gene for erythroid cells. In this plot, each dot is a cell. X-axis shows the pseudotime. Y-axis shows the expression of the HBA2 gene. We can see that this gene has an overall increasing pattern along the pseudotime, which roughly agrees with the pattern with the process of hematopoietic stem cells differentiating into erythroid cells. However, this gene has an unexpected decrease of expression at the end of the pseudotime, which does not quite agree with the non-biology. So why does that happen? If we revisit the minimum spanning tree, we can see that it takes a turn here and points down to the cells with low expression in HBA2, instead of pointing to the top right corner to the cells with high expression in HBA2. This happens because Monocle tries to link each individual cell. Because there are thousands of cells, the spanning tree can be highly variable and unstable, and the pseudotime trajectory may not reflect the true biological process. Here shows a toy example to help you better understand. Each dot here is again a cell, and the true biological time runs down, top down vertically, and the plot on the left and right give 2 possible solutions to the minimum spanning tree because they have the same total edge weight. Now suppose there's a small measurement error added on the cell, which is quite common in single-cell sequencing data, and pushing it away from all the other cells. Then the plot on the left will become the only solution to minimum spanning tree, and Monocle will pick it to construct the pseudotime. However, the plot on the left does not quite agree with the true biological time, which does not allow time to go backwards. And in fact, the plot on the right agrees better with the true biological time. Unfortunately, Monaco picks the wrong one to construct the pseudotime. To address this issue, we developed a new method called T-scan, and it is published on nucleic acid research in 2016. And after doing T-scan pseudotime reconstruction, the expression of the HBA2 gene increases monotonically along pseudotime, which better agrees with the known biology. So how does T-scan work? In Monaco, we have seen that the high variability causes some problems. In statistics, how do we deal with high variability? We often use bias-variance trade-off. Bias-variance trade-off is a well-known concept in statistics and machine learning literature. However, no one has applied this concept before in the pseudotime and spanning tree inference, and we are the first to apply this concept to construct the pseudotime trajectory, and this is our unique contribution. Specifically, T-scan first groups cells into clusters, and then builds a minimum spanning tree to link the cluster centers, and finally project each individual cell onto the minimum spanning tree to form the pseudotime trajectory. This process brings a little bit bias because the cluster center may not represent the true state of each individual cell. However, it greatly reduces the variability of the spanning tree, thus leads to a more accurate and robust pseudotime trajectory inference. And we call this method cluster-based minimum spanning tree. It turns out that the bias-variance trade-off is in favor of the final performance, as we can see in the HBA2 gene example. Another unique contribution of our work is that we're the first to develop methods to systematically evaluate the performance of different pseudotime analysis methods. For example, we take advantage of time course experiments where single-cell RNA-seq are done in different time points. And we design the score to measure the consistency between the experimental time and pseudotime, given that we perform the pseudotime pretending we don't know the experimental time. And the better pseudotime analysis method should have a higher consistency score. Other metrics include the robustness of pseudotime and the ability to recover non-differential genes. In all these analyses, T-scan has the best overall performance. In recent years, there's a third-party benchmark paper comparing many different pseudotime analysis methods using a variety of different benchmarking scores. In this plot, each dot is a method. X-axis shows the time of the publication. Y-axis shows the overall benchmark score. And the right line here traces the best method over time. T-scan has the best performance at the time of its publication. After that, many other pseudotime analysis methods were published, including an updated version of Monaco, but T-scan still has the best performance until Slingshot, which was developed by Sandra Dudu's group at UC Berkeley. However, Slingshot builds upon T-scan, and its first step is to perform the cluster-based minimum spanning tree, and then use the principal curve to improve the performance. We are happy to see that Slingshot improves T-scan, and we are also happy to see that we are able to contribute a generalizable principle to the field of pseudotime analysis, which becomes the building block of future methods. You may have noticed that the number of cell clusters may greatly affect the structure of the tree. In fact, a large number of cell clusters may help the tree to capture some local structures, but may also induce a higher variability. In our original publication, the number of cell clusters is determined either based on some model-based assumptions or given by the user. And we are working on improved version of T-scan, where the number of clusters is chosen automatically using some cross-validation procedure. We have applied T-scan in several collaboration projects. In one of the collaboration projects with Dr. John Ware's group at University of Pennsylvania, we studied the process of T-cell exhaustion, where T cells loses their effective functions during chronic infections or cancer. In the study, we applied T-scan and pseudotime analysis and identified a transcriptional network where a key factor called TCF1 may regulate the other genes to together initiate the T-cell exhaustion process. In another ongoing project, we built a brain cell atlas by integrating data from many publicly available single-cell RNA-seq studies. And we used T-scan to build a differentiation trajectory from stem cells into neurons, oligodendrocytes, and astrocytes. These are all common cell types in brain. And this differential process is something people already know. We are collaborating with doctors at Johns Hopkins who are interested in glioblastoma and are collecting single-cell RNA-seq data for glioblastoma patients. And we integrate the patient cells onto the atlas and the pseudotype trajectory to infer the origins of the tumor. For example, the location of the cells on the trajectory from stem cells to neuron can be used to determine whether these cells are more likely to be originating from stem cells, from neurons, or from some intermediate cell types. An initial analysis suggests that the distribution of the cells on the trajectory is actually associated with the location of the tumor in the brain. For example, for patients whose tumor coming from the frontal lobe in the brain, the distribution tend to tilt to neuron. And for patients whose tumor coming from occipital lobe in the brain, the distribution tend to tilt to stem cell, suggesting different tumor mechanisms for tumor from different parts of the brain. We are now collecting more single-cell data sets and performing more systematic analysis to further validate this hypothesis. A future research direction is to develop methods to more systematically integrate patient cells onto the atlas and to associate with patient outcome. To wrap up the first part of my talk, we developed a new pseudotype analysis method called T-scan by using the bias variance trade-off concept in the pseudotype tree inference, which significantly improves the accuracy of the tree construction. And we have also developed methods to systematically evaluate the performance of different pseudotype analysis methods. In pseudotype analysis, we identify genes that changes over pseudotime. And we may further ask why these changes happen. This brings us to the second part of my talk, where single-cell regulant data may come into play. Unfortunately, single-cell regulant data is highly sparse and noisy. So we developed a new method called SCATE that enhances this highly sparse signal by adaptively integrating information from multiple sources. Before introducing my method, I'm going to introduce some basic biology. There are many mechanisms regulating the gene expression process. One important mechanism is called chromatin remodeling. In our cells, the DNA are wrapped around these small balls called nucleosomes. And if the nucleosomes are tightly packed together, the chromatin is closed. And a special type of protein called transcription factor, or TF, cannot bind to the DNA and regulate the expression of the gene. If the chromatin goes through structural remodeling, these small balls or nucleosomes are no longer tightly packed together. And the region of non-coding DNA called cis regulatory element, or CRE, get exposed, and the transcription factor can bind to the CRE and regulate the expression of the gene. Different transcription factors, or TFs, tend to recognize specific DNA sequence. And if we know a cis-regulated element is in open chromatin region, we can computationally identify the type of transcription factor binding to the CRE and regulating the expression of the gene, thus explaining why this gene is expressed or not. DNA-seq and ATAC-seq are two state-of-art technologies to measure the level of chromatin openness, or chromatin accessibility. These two technologies will produce a lot of reads for open chromatin regions and far less reads for closed chromatin regions. DNA-seq requires millions of cells, and ATAC-seq requires hundreds to thousands of cells, so they cannot be used to study the variability across single cells. To address this problem, people developed a new technology called single-cell ATAC-seq. Single-cell ATAC-seq is able to measure the chromatin accessibility in each individual cell. Unfortunately, the signal from single-cell ATAC-seq is highly sparse and noisy. This plot shows the signal of bulk DNA-seq and raw single-cell ATAC-seq for the same genomic region and for the same cell type. Compared to the relatively continuous signal from bulk DNA-seq, the signal from single-cell ATAC-seq is highly sparse and nearly binary. For example, it does not have any signal at all for the red box and only has one read for these two regions. This happens because the diploid genome only has 2 copies of DNA. And single-cell ATAC-seq only has a chance of 1 or 2 to capture the open chromatin regions. However, molecular events such as transcription factor binding to DNA is a temporal stochastic event. And such highly sparse and nearly binary signal cannot accurately describe the probability of these molecular events happening. Thus, we need to find a way to enhance the signal. So we developed a method called SCATE to do this. After the signal enhancement, the signal looks much more continuous and better represents the probability of the molecular events happening, which is characterized by the bulk DNA-seq data. So how does T-scan work? How does SCATE work? Before introducing our method, it's useful to go through the existing methods. There are 2 types of existing methods. The first type of method mitigates the sparsity of the signal by aggregating information across predefined sets of CREs, which are either co-activated or share some biological features. These methods include ChromeVar, Brockman, and SCRAC developed by us. Because these methods aggregate information across CREs, they may lose CRE-specific information. Also, because the CRE pathways are predefined, the level of information aggregation is the same for all the single-cell ATAC-seq data. The second type of methods mitigate the sparsity by aggregating information across cells. These methods include DrSeq2 and Cicero. Because these methods aggregate information across cells, they may lose cell-specific information. Also, more importantly, for cell population with small number of cells, the sparsity issue is still very severe, even after the signal aggregation. So ideally, people want to study the activity in each CRE and in each cell. So it requires us to pull information across as few CREs and cells as possible. However, the high sparsity and noisy nature of the data requires us to aggregate information across more CREs and cells. So there's a dilemma. Because we cannot solve these 2 problems simultaneously, what we can do best is to find an optimal trade-off. Unfortunately, no existing method tells us where this optimal trade-off is. SCATE tries to find the optimal trade-off by adaptively aggregating information across CREs. Intuitively, for cells with less information, SCATE pulls information across more CREs to increase the robustness of the estimate. For cells with more information, SCATE pulls information across less CREs to keep the CRE-specific information. This idea of adaptively aggregating information across CREs is a crucial part of SCATE and our unique contribution. SCATE also pulls information across cells by first grouping cells into clusters, and then pulling the reads across all cells within each cell cluster to form the pseudocells, and finally run SCATE on these pseudocells to generate the enhanced signals. Another unique feature of SCATE is that it utilizes huge amounts of publicly available data. This plot shows the signal of bulk DNA-seq of different cell types. Each row or track here shows the signal of one cell type. What can we observe? We can observe that there are some consistent patterns. For example, this peak has consistently higher activity, and this peak has consistently lower activity across all these cell types. This means that for a new cell type, we can use the average activity across all these cell types to roughly predict its chromatin accessibility signal without even doing a single cell experiment. More importantly, this information is often not contained in a single cell ATAC-seq data. For example, the first track here shows a signal of single cell ATAC-seq data of a randomly chosen cell. We can see that it does not have signal at all for these two boxes. However, this averaging approach also has its own problem. That is, the average signal is the same for all cell types and does not contain cell type specific information. Based on all these observations, we developed SCATE that adaptively pulls information across co-activated CIEs from large amounts of publicly available data and from similar cells. The goal of SCATE is to mitigate the high sparsity of the single-cell ATAC-seq data while retain the CIE and cell-specific information as much as possible. Now I'm going to introduce some technical details of SCATE. We start from the observed single-cell ATAC-seq recount Y. Our goal is to estimate the true activity mu. We assume that log true activity, log mu, equals baseline mean M plus baseline standard deviation S times cell-specific factor delta. Here, M and S are estimated for each CIE across all cell types in the historical bulk database. Delta captures the cell type specific information that cannot be captured by M and S because M and S are the same across all the cell types. Delta here is unknown. It needs to be estimated from the single-cell ATAC-seq data. To mitigate the high sparsity of the single-cell ATAC-seq data, we further use the bulk database and group CIEs into clusters. And we assume that delta is the same across all CIEs within each cluster, and we call it cluster factor beta. A unique feature of SCATE is that the number of CIE clusters is not determined a priori, but rather we use a cross-validation procedure to determine the optimal number of CIE clusters based on the single-cell ATAC-seq data itself. Equivalently, we can use a low-rank representation of a binary cluster membership matrix X multiplies the cluster factor beta to replace the high-dimensional cell-specific factor delta. Finally, notice that the true activity is based on the bulk DNA-seq data, and the observed recount is based on the single-cell ATAC-seq data. So we need to normalize between the two sequencing platforms. To do that, we assume that true activity mu is distorted into mu-SC in single-cell ATAC-seq through an unknown parametric monotone spline transformation function H. And the observed recount Y is further generated from mu-SC through a Poisson distribution. Now, to estimate the parameters in H, we again use a bulk database and identify a list of CIEs with low variability across all the bulk cell types. And we assume that these CIEs have the same activity across all the cell types, including the cell type in single-cell data. So we can use the average information across all these cell types to serve as a surrogate for mu. Because we have already observed Y, we can then estimate the parameters in H. Putting everything together, we start from the observed single-cell ATAC-seq recount Y, and our goal is to estimate the true activity mu, which can be decomposed into two parts. The first part, M and S, that can be estimated from the historical bulk database, and the second part, beta, that can be estimated from the single-cell ATAC-seq. Here shows the same mathematical formulation of the SCATE model, as well as some technical details. I'm not going to go through this, but you are welcome to refer to the SCATE publication. Now, to benchmark the performance of different methods, we calculate the correlation between bulk DNAs and single-cell ATAC-seq data for the same cell type. Here, x-axis shows the signal of bulk DNA-seq, which kind of acts as a gold standard here. Y-axis shows the single-cell ATAC-seq signal, either from SCATE or from the raw signal. Each dot here shows the signal of a 200 base pair genomic bin. We can see that the signal from SCATE is much more continuous and has a higher correlation compared to the raw signal. Next, to evaluate whether SCATE can keep the variability between single cells, we perform a differential correlation analysis. We calculate the differential signal between two cell types for bulk DNA-seq data and the differential signal in single-cell ATAC-seq data for the same two cell types. And we calculate the correlation between these two differential signals, and we call it differential correlation. Here, x-axis again shows the differential signal for the bulk DNA-seq data, and y-axis shows the differential signal for the single-cell ATAC-seq data. We can see that, again, SCATE has a higher differential correlation compared to the raw signal. Next, we include the baseline mean, which is the average information across all the historical bulk DNA-seq data. We can see that although it has a relatively high correlation with the gold standard bulk, although not as high as GATE, it does not have any cell type specific information at all, so it totally fails the differential correlation analysis. Finally, we include an example of existing method that only pulls information across CREs. This method has a lower correlation and differential correlation compared to SCATE. We have also systematically compared the performance of different methods with different number of cells pooled and in different data sets. This analysis includes the correlation and differential correlation analysis we have just seen, as well as the performance of p-calling, where we need to find the regions with high signal. SCATE, which is marked as a red line, has the best overall performance. More importantly, SCATE substantially improves upon the raw signal. For example, in the correlation analysis with one cell, SCATE has an approximately 250% increase of performance over the raw signal, which is quite amazing. As an application of SCATE, we can use the SCATE enhanced signal to predict the transcription factor binding sites. For example, we can use the motif information of ELF1, which is one type of transcription factor, and scan the whole genome to find the potential binding sites of ELF1. F1 potential binding site also has high SCATE enhanced signal. We can predict this region as an active binding site for ELF1. We use the bulk ChIP-seq data for the same transcription factor and for the same cell type as a gold standard. In this case, SCATE with one cell reaches an area under sensitivity FDR curve of around 0.65, which is again 90% increase of performance overall. The prediction method used by SCATE is a relatively simple one. In fact, we have developed a much more powerful predicting method in a community challenge called ENCODE Dream Challenge, and I was the leader of our team. In this challenge, we use massive amounts of publicly available bulk RNA-seq data, bulk DNA-seq data, and DNA sequence information to predict the transcription factor binding events for many different transcription factors in many different cell types. And here shows the overall performance, and our team ranks 4th out of all the 50 participating teams. So the next step will be to combine SCATE enhanced signal with this more powerful prediction method to explore the possibility of predicting transcription factor binding events in single cell. To wrap up the second part of my talk, we developed a new method called SCATE that adaptively integrates information across CREs and cells to enhance the highly sparse and noisy single-cell ATAC-seq signal. In SCATE, we have demonstrated that it is possible to recover the chromatin accessibility information in single cell. Unfortunately, for most of the single-cell data, only single-cell RNA-seq data is available, and only a small proportion of single-cell genomic data contains the single-cell ATAC-seq information required by SCATE. So it raises the question is whether we can use the relatively rich single-cell gene expression data to infer the much less available single-cell regulin data. This motivates us to develop a new method called BIRD that predicts the chromatin accessibility with gene expression. BIRD is a supervised precision model that builds upon a large collection of cell types with both bulk DNA-seq and bulk RNA-seq. And for a new cell type coming in, either bulk or single cell, BIRD is able to predict the chromatin accessibility for the new cell type. And the predicted value can be used for downstream analysis, such as the prediction of transcription factor binding sites. Here shows the signal of single-cell ATAC-seq experimental data, the BIRD predicted signal for one single cell, and the bulk DNA-seq data. We can see that the BIRD predict signal looks much alike the bulk DNA-seq data, and it's much better than the single-cell ATAC-seq data. We have also conducted systematic evaluation, comparing using different data sets and with different evaluation criteria. And BIRD is able to largely recover the chromatin accessibility signal even for one cell. So with BIRD, we can largely increase the value of existing single-cell or bulk RNA-seq data, as well as providing a rich resource to study the regulatory activities in many different biological contexts. Building upon all these methods, we also developed Reason, which is a regression and differential analysis framework with multiple samples. Reason addresses the multi-level variance structure that is increasingly popular for recently emerged large-scale single-cell genomic data, and works for both single-cell RNA-seq and experimental or predicted single-cell ATAC-seq signal. For time constraint, I'm not going to introduce the details of Reason here, but you are welcome to discuss with me after the talk. We are applying all these methods in a neoadjuvant immunotherapy clinical trial for non-small cell lung cancer patients. This is a collaboration project with Dr. Drew Paddock and Dr. Kelly Smith at the Bloomberg Kimmel Center for Cancer Immunotherapy at Hopkins. In this clinical trial, there are patients who receive or do not receive the immunotherapy. Within patients who receive the immunotherapy, there are patients who respond or do not respond to the immunotherapy. For each patient, single-cell genomic data is collected at different time points for different cell types and from different data modalities. For example, they are using a recently developed method called manifest assay to trace the mana-specific T cells. Here, mana stands for mutation-associated neoantigens, and these T cells are likely to recognize the cancer cells. And we can use the TCR barcodes from these T cells to track in the paired single-cell RNA-seq and TCR-seq data and study the gene expression profiles for these mana-specific T cells. One key research problem in this study is to identify molecular signatures differentiating different groups of patients, for example, responders and non-responders. To do that, again, we map all the cells to a low-dimensional representation and group cells into clusters. And we have an expert who annotates the potential cell types for each cluster. Within each cluster, we can run Reason to identify differential genes which have differential activity between responders and non-responders. We also run BIRD to predict the transcription of the chromatin accessibility within each cell, and again use Reason to identify the differential cis regulatory elements. The hope is that we can use these differential signals to identify new mechanisms that differentiates the responders and non-responders. For example, we can potentially find new receptor ligand pairs such as PD-1, PD-L1, and probably that can be work for future drug treatments. Another possibility is that we can use these differential signals to identify new biomarkers that can predict the patient early outcome. These projects also brings countless future research directions. The one direction is to develop methods to integrate single-cell data from multiple samples. For example, we can integrate the immune repertoire across different patients and for different antigens. We can develop methods to identify genes that have differential activities along the pseudotype and between different groups of patients. We can also develop methods to identify genes that not only have differential mean, but also has differential variability across different groups of samples. The second direction is to develop methods to integrate single-cell data from multiple modalities. For example, we can predict other types of epigenomic signals, such as DNA methylation, histone modification, and DNA 3D structure. We can develop methods to better analyze the paired single-cell RNA-seq and single-cell TCR-seq data. We can combine the CRISPR-based perturbations with single-cell RNA-seq and single-cell ATAC-seq to better infer the gene regulatory networks. We can combine the spatial information from, for example, 10x Visium spatial technology to study the spatial transcriptomics and spatial epigenomics. Finally, using all these features, we can build highly scalable machine learning methods such as deep neural networks to predict the patient outcomes and decipher gene regulatory programs. So this concludes the main part of my talk. I'm going to go to the acknowledgements part. The first person and most important person I want to thank is my CDS advisor, Dr. Hongkai Ji. And I've been working with Hongkai for 7 years. And actually, I can still remember the first day, 7 years ago, when I came to Hongkai's office and discussed with him the potential research projects I can do. And in these years, I've not only learn from him the necessary skills and knowledge in the field of statistics and computational biology, but also the way of independent and critical thinking, as well as how to efficiently communicate the results to the broader scientific community. I'm extremely lucky to have him as my PhD thesis advisor. I'd like also to thank the members of my thesis committee and the current lab members in Hongkai's group, Dr. Wenping Ho, Dr. Weixiang Zhou, Weixiang Fang, and Boyang Zhang, and all the former lab members who have greatly helped my various research projects. I will thank all the faculty members in the Department of Biostatistics. I'd like to especially thank Dr. Marie Dina West, Dr. Stephanie Hicks, Dr. Nija, and Dr. Meichen Wang for providing huge help in my faculty position search this year. I'd like to thank all my wonderful collaborators at Johns Hopkins School of Medicine, Dr. Drew Padol, Dr. Kelly Smith, Jaja and Justina, Dr. Chitam Batagoda, Christina, Dr. Janice Taub, and Sneha. I'd like to thank Dr. Andrew Feinberg, Veronica and Michael, Dr. Sean Taverna, and Dr. Kimberly Stephens. I'd like to thank my wonderful collaborators at Johns Hopkins Rogoff School of Public Health, Dr. Xiaobing Wang, Dr. Xuemei Hong, and Dr. Guoying Wang. I will thank my collaborators at the University of Texas at Austin, Dr. Stephen Vaux, and Rachel and Christine. I'd like to thank collaborators at University of Pennsylvania School of Medicine, Dr. John Wei and Zoe Chen. Finally, I'd like to thank Dr. Fang Han. He used to be a PhD student in our department, and he is now a faculty member at Department of Statistics and University of Washington. Finally, I'd like to thank my family, my wife, and my parents who have provided huge support during the full course of my graduate studies. Thank you. And here is some methodology work and some collaboration work during my PhD studies. Thank you very much for your attention, and I welcome any question.
All right. So thank you very much, Jason. So now we have time for a couple of questions. So if you have questions, you can unmute yourself and ask, or you can raise your hand.
Jason, that was really wonderful. Thank you so much. And I have to say, I think as the lone immunologist, I think followed a lot of what you were saying. So I think that's a real testament to, you know, your ability to make it as an independent investigator in the broader scientific community. So excellent job. So kind of the obvious question then, is there a reason to ever do single-cell ATAC-seq if your methods are able to kind of infer chromatin accessibility?
Thanks, that's a very good question. There's still value in doing single-cell ATAC-seq. So because what we use is a kind of historical bulk data, and in many situations, this data set may not cover the biological system of interest in the specific study. For example, in your study, there's like a lung cancer sample from a specific kind of tumor. And this kind of sample may not be already included in our database. So this signal may not already be captured by our historical bulk data. Thus, this is kind of an additional information from the experimental data that if only use our prediction method may not be captured. So there's still value. And I think as actually has been already demonstrated in the BIRD paper, that the predicted signal from BIRD is able to kind of enhance the signal of the single-cell ATAC-seq data if you have paired single-cell RNA-seq and single-cell ATAC-seq for the same biological system. So there's, of course, there's still value in doing single-cell ATAC-seq because the prediction is not perfect.
Gotcha, thank you.
So Karen has a question. Karen, maybe you can. Yeah.
Jason, congratulations. That was absolutely wonderful. And I thought the insight into the biological processes that you've provided through statistical thinking is really spectacular. So first of all, thank you for that. My question is actually a lot like Kelly's. It was about SCATE. And You focused, I thought, quite a bit on validating your methods with regarding to the single cell ATAC with respect to bulk ATAC. And I would love to hear you talk a little bit more about the situations under which the bulk is an appropriate gold standard, as I would expect that one would do single cell precisely because there could be variability even within the same cell type. So thank you.
Yeah, thanks, Karen. This is actually an excellent question. And I would answer in this way. So first, the bulk data, the bulk gold standard is not ideal. So because you have mentioned the single cell data itself has variability, and the bulk data may not capture this variability. But unfortunately, the current technology, we don't have better gold standard than the bulk DNA-seq data, because actually we need to know the signal for the single cell itself. But the best technology we have now is already single cell ATAC-seq. So actually we don't have a better technology. So bulk DNA is kind of a suboptimal, but the best gold standard we can have already now. And to answer the question that whether the bulk can be used to measure the variability across single cell, actually we have demonstrated that there's a differential correlation analysis where we have demonstrated that SCID has the best performance as well in the differential correlation. So here, differential correlation actually already kind of measures whether the method can capture the variability across single cell. Because if the variability can be captured, then the differential signal between two cell types should also be captured. So I would I would like to say that a differential correlation actually kind of already takes care of this kind of disadvantage of the bulk gold standard.
That's really helpful. Thank you.
Good question. So congratulations, Jason. It's wonderful talk. So you demonstrated beautifully for the pseudo-time analysis, and just curious a little bit more how you could potentially extend it. I'm aware you guys have tried some of those in Hong Kai's lab on other dimensions, on spatial dimensions, or even further dimensions. Could you please make some comments about that?
Yeah, so that's a very good question. And what we are doing now is, we are thinking of more like methodology aspects of further extending the current T-scan method. So for example, as I mentioned in my talk, we in the original publication, we choose a number of cell clusters using more like a relatively more ad hoc procedure. And we will develop a better method in the future to systematically choose a number of clusters using, for example, a cross-validation procedure. And what we are actively working now is to do the pseudo-time analysis with multiple samples. For example, if you have patients from responders and non-responders, and there are multiple patients within each group, then can you identify genes that show differential activities between these two groups along the same pseudo-time? And that's more like an emergent question, because now such kind of single-cell RNA-seq data just start to emerge, and you don't have this kind of data in the past due to the cost of the single-cell technology. And in terms of your question whether you can kind of extend it in other dimensions, such as spatial, I think definitely you can do that. So I would imagine, for example, you can build a pseudo-time trajectory for different parts of the brain, for example, and then compare, for example, whether there's difference between the pseudo-time trajectory across different locations of the brain, something like that. And also, for example, what we can do is like in the immunotherapy aspect, there are like T cells, and we can use the TCR sequencing to track the different clones. And for example, we can, if you have time course data, we can track the temporal changes of gene expression for each TCR clones. And some clones are very interested. For example, they recognize the cancer cells. And that's another way that can bring other dimensions of the data into the pseudo-time analysis. Thank you.
All right, so there are other questions.
Thank you, Jason. That was a wonderful, wonderful presentation, and congratulations. I'm so impressed with all of your work, and so happy to hear that you've landed another faculty position, and I'm sure you're going to go on to do great things. So my question's about imputation. So I'm less familiar with imputation in the context of AttackSeek. Is that happening, or do you think that would be helpful, or are there approaches that you're aware of that people are using?
So if you are talking about imputation based on the peaks, so for example, you can first pull all the single cells as a pseudo bulk and then call peaks on the pseudo box. And then you have, for example, a peak by cell matrix where each row is a peak called in the pseudo bulk sample and each column is a cell. And then the number in this matrix is the number of reads. Then I think there's a method already. There's a method called scale, SCALE, that uses a deep learning framework to do that. And also, I think other methods I mentioned in my talk, I don't know whether they can exactly do imputation, but I believe some of them can do imputation as well. The so-called imputation, but probably they don't mention imputation, the word imputation in that paper, but probably they can do similar things. And if you are talking about the whole genome kind of analysis, then I think our method SCATE is probably the first one that can do this kind of analysis. So I think almost all the methods now, they focus on the peaks that are caught in the pseudo bulk sample. So apparently you have a large proportion of the genome that is not covered by these peaks. And actually SCATE, because it takes advantage of the historical bulk data, so it actually can recover the signal for almost all the cis-regulated elements for the whole genome. So that's a difference of SCATE, compared SCATE to other existing methods.
Thank you.
All right, if there's no more questions, we're going to end our public portion of the defense. And committee members, please stay. And all others, thank you very much for attending. And I'm going to unmute all, so in case you want to clap and congratulate again.
Good job.
Thank you very much.